Hey yo, what's up, YouTube? It's your boy, Josh Abraham. It's your boy Cracker Tom, innit? It's your boy Josh Abraham, innit? Innit? Well, yeah, I just found out yesterday when I went to see the movies, and I had a conversation with some of my friends, and he just told me that there's one more PDF. And that's how I found about these. So I forgot about these parts of the question. I need to make sure I know this. Describe the function of E. coli. Okay, let's see. Design of origin of replication. Selectable genes, multiple cloning sites. Origin of replication. Okay, you got that. Selectable markers, you got that. Multiple cloning sites, you got that. Reporter gene, small size. Okay. GPT got yeah. GPT got this time. Origin of replication, selectable marker, reporter gene, small size. So yeah, both are similar. Mm, got a request from a friend. Mm, oh shit, I have to make some difference, some changes. Just a minute, look, YouTube. Just a minute. Uh, Dude, I just woke up and I feel so tired. What the? No, I need to. I believe it's good. It's yeah, that's okay. This part is yeah. So people, so YouTube, I'm really sorry to tell you this, but I need a 15 minutes break. I don't know, my mind is so tired right now. So I'll be right back. 15 minutes. Hey Siri, set a timer for 15 minutes. 15 minutes. I'll be right back. Just stay strong. Stay strong. Stay strong. Someone was on the chat. I'm sorry, man. Daniel Carnero, are you still here? I took a 15 minutes break, man. I'm really sorry, man. I was not here. How long has this been going? I started streaming 59 minutes. I accidentally slept. I'm really sorry. It should have been like, it shouldn't have been. Oh, Noor, you're still here. I appreciate it. Shout out to Noor. I accidentally slept more than 15 minutes than I should be. I should have been sleeping for 15 minutes. Noor, what's up? How you doing, man? Even OBS, uh, I'm guessing it's time for Undertale. Yeah. I mean, if there's an engaging viewer, you got it. Undertale it is. Oh, yesterday I did not post the Undertale gameplay. I forgot about doing that. So 15 minutes of Undertale, let's go! Even though I did not study shit. Elder Scrolls. Elder Scrolls. Have you completed the game, Undertale? Hey Siri, set a timer for 15 minutes. Okay. We get it. So, yesterday, if I remember, it's about our sports. I thought we might celebrate your arrival. I want you to have a nice time leaving. I know. I actually went through this. I saw my room. So Noor, or N hour, I don't know. How do you pronounce your name? That's it. Is this your first playthrough? Yeah, this is my first time playing Undertale, and this would be part 4, I believe. I'm sorry, ma'am. I don't wanna. You'd rather stay up and chat with me then? I want you to know I'm glad I have someone here. I want to show you my favorite bug hunting sport. Curriculum for education. Always wanted to be a teacher. I'm sorry, woman. Did you want something? What is it? I'm sorry. What? This is your home. Like to hear about the book I'm reading? It's called 72 Ways. I did hear a bit about the book. Did you know that snails sometimes look their... What did I press? You pronounced my name right. Okay, Noor. So, have you completed the game? No, stay here. Stay here, woman. She just left. You know what it is? It's the basement. Oh shit. You wish to know how to return to home, do you not? Ahead of us lies the end of the ruins. One way exit to the rest of the underground. I'm going to destroy it. No one will ever able to leave again. Now be a good child and go upstairs. I'm I'm done. I've done everything. Okay, so you completed the game. Actually started with Undertale, the Delta Rune, but someone on Twitch told me that it's better to start with Undertale. Every human that falls down here meets the same fate. I have seen it again and again. They come, they leave, they die. You naive child. If you leave the ruins, they Ascor will kill you. I'm only protecting you. Do you understand? Go to your room. I'm sorry, man. Do not try to stop me. This is your final warning. I mean, what do you expect? You want to leave so badly. You are just like the others. There's only one solution to this. Prove yourself. Prove to me you're strong enough to survive. How? Oh shit. Gotta act. Talk. Well, that's a bummer. Well, we gotta fight. Bullshit. Dude, I'm dead at this point. Let's see. Oh. Okay. Sure. Oh, interesting. Now I feel sad that she's letting me win. Now what if I die? I could just kill myself. No way. I can't kill myself. So, Norm, have you completed under the Delta Room? I'm sorry. You are stronger than I thought. Listen to me, small one. If you go beyond this door, keep walking as far as you can. Eventually you will reach an exit. I'm sorry, man. Ascor. Do not let Ascor take your soul. This plan cannot be allowed to succeed. Be good, won't you? Yeah? My child. I didn't mean to. I could come back, you know. I completed it, but I haven't done everything in it yet. So, you have done everything on Undertale. God damn. I Hope you like your choice. After all, it's not as if you can go back and change fate. In this world, it's kill or to be killed. That old hag thought she could break the rules. She tried so hard to save you humans. Boy, Benny. I love this sunflower. Motherfucker, bro. This motherfucker. It's the sun. Are you aware of the multiple endings? That's the game? What? No way, I mean, it's a new place, right? Yeah. Okay, so, yeah, I do know that there are mainly 3 endings, and the rest of the endings are actually part of the 3 endings, right? Oh. 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 It's a big man. It's a big ball. Human. Don't you know how to greet a new pal? Turn around and shake my hand. Okay. That's the hand? What level are you? Oh, it's the sands. The old whoopee cushion in the hand trick. Okay. It's always funny. Anyways, you're a human, right? That's hilarious. Sans the skeleton. Actually supposed to be on watch for humans right now. But, you know, I don't really care about capturing anybody. Now my brother. Papyrus? What level are you? Don't tell him about the endings, just let him have his... Vassinic 12, what's up? It's been some time. He's a human hunting fanatic. Hey, actually, I think that's him over there. I have an idea. Go through this gate thingy. Go right through. My bro made the bars too wide to stop anyone. Wait behind that convenient shaped. You can be more quick. Oh. Sup, bro? You know what? Sup, brother? It's been 8 days and you still haven't recalibrated your puzzles. You just hang around outside your station. What are you even doing? It's been a while. Yeah, it's been a while. So, yeah, I think I just started the game. I was really interested in seeing this character, Sans. Staring at this lamp, it's really cool. Do you want to look? No, I don't have time for that. What if a human comes through here? I want to be ready. I will be the one. I must be the one. I will capture human. Then I the great papyrus, will get all the things I utterly deserve. Respect, recognition. I will finally be able to join the royal guard. People will ask to be my friend. I will bathe in the shower of kisses every morning. Maybe this lamp will help you. Since you're not helping, you lazy bones. All you do is bondogo. You get lazier and lazier every day. Hey, take it easy. I got a ton of work today done. A skeleton. Get it? A skeleton. Your PFP looks familiar. Have I seen you in different Undertale stream? Oh, okay, I thought it was about me. Sans, come on, you're smiling. I am, and I hate you. Why does someone as great as me have to do so much just to get some recognition? Wow, sounds like you're really working on yourself down to the bone. I mean, Nathan Reed, banana. Banana. I will attend to my puzzles. As for your work, put a little more backbone into it. Okay, you guys know each other or somewhere. Okay, you can come out now. Whew. Oh boy, that was a hard one. That's it? Okay. Uh, you ought to get going. He might come back. If he does, you'll have to sit through more of my hilarious jokes. Hell yeah. Can I do something? Nope. Actually, hate to bother you, but can you do me a favor? I was thinking my brother's been kinda down lately. He's never seen a human before, and seeing you might just make his day. Don't worry, he's dangerous. He's not dangerous even if he tries to be. Thanks a million, I'll be up ahead. Okay. Nathan Reed, hi Mason. What's up, people? I'm glad you people get to know each other on this stream. It only happens on this stream. Save, yeah, fuck yeah. Okay, I'm on level 5. Use the box. Okay. Tough love. The music's pretty good. This is a box. You can put an item inside or take an item out. The same box will appear later, so don't worry about coming back. Sincerely, a box lover. You know what? I wanna fight this thing. Better not snowflake out. Okay, it's been 15 minutes. Oh shit. 15 minutes and... Whoa! The level up is working! Okay. Okay, I'll continue later. Well, that's it for today. Undertale for today. And let me tell you people, the game is getting more interesting. I did save, yeah, I did save. That was I really like the Sans and the brothers. It's really interesting. So, what are you up to, people? How we doing? And yeah, my exam is tomorrow, and I just realized there's one more PDF I have to look into. And I'm realizing this the day just before the exam. How stupid can you get? It's hilarious. Get in. Just get in. I'm guessing everyone left the stream, or is my stream a bit slow? Good luck with your exam. I appreciate it, Nor. I appreciate it. Oh, I have been playing games, to be honest. Okay. Nathan, what type of games are you playing other than Undertale or Deltarune? Describe the required functional elements of typical vector cloning. required for autonomous replication of E. coli, regulation of last bit copy number, initiation of DNA replication, required for autonomous replication, regulation of initiation of DNA replication, determines copy number, regulation of plasmid copy number. It's a bit different. What? We're still here. I appreciate it, man. I really appreciate it. So, what are you guys up to? Are you playing some sort of games? Uh, I mean, how is the Roblox going, Masonic 12? Cause I saw some of the videos that you tried posting. Just chilling, honestly. Okay. I mean, yeah, it's a good day to chill. It's a Tuesday. I think I have to head to work in about 12, 1, 1:30. Yeah, I have to be out at around 1:30. Now it's 11:32, so 12:30. 2 hours. I have to. These 2 hours, I gotta make sure I do it. The games here and there mixed with YouTube. I do plan doing some more Deltarune though. Okay. I started playing Deltarune, but for me, I felt got some loyal viewers. I really did. I appreciate it, people. I really appreciate the support, cause it's not always bad things out there. There are real people who want to support, and I think it's a really beautiful thing. Yeah. So, what was I saying? Yeah, for Deltarune, I felt the game is kinda slow, maybe compared to Undertale. Undertale, within the first three episodes, it started picking up the pace. At least for me. Masonic brought me here. Oh shit! I appreciate that. I really appreciate that. So, are you a friend of Masonic, or are you part of his channel? How do you guys know each other? I mean, I don't know if it's a private question. I'm sorry if it's a private question. Uh, ampicillin resistance allows plasmid to replicate independently. And it's a fire stream. I appreciate it, man. I mean, so the basic. I don't. You're the first time here, right? So I'll explain the basically what's happening here. A friend, he's been in few of my streams. Okay, so I really appreciate that. So Nathan Reed, if you're our first time here, so basically last time December, I was in a really, like, my education part was not going really well. My studies was at the bottom. It was at the bottom, and I just thought maybe I could stream first time. So maybe I could stream what I'm studying and maybe play some games in between. Like, yeah. So when I first started the streaming, at that time I also bought a game called Lice of P, which I did pay a bit amount. So I thought maybe if there's an engaging viewer, I will play the game, if only if there's an engaging viewer. That is, only if the stream is going on. And oh boy, this computer does not handle a, I don't know, Lysipe is a AAA game, but a pretty graphically intensive game and stream together, this computer does not handle. So that's how I changed to. I bought the game Undertale. It was at offer. It was for €2 or something. Do you know you can run by holding X in Deltarune? I thought it was Shift, or is it just for Deltarune? Is it applicable to Undertale? Yeah. So I was saying, and that's how I changed to Deltarune. And when I was on a stream once on Twitch, someone told me that instead of playing Deltarune, it's better to start Undertale. That's how I got into Undertale. But before that, I did hear about this game. I heard that it's a really cool game, the Undertale. It does have a mark on the gaming platform. Shift or X? Yeah, I think it's Shift. At least for my computer was Shift. But the worst thing was I did not notice. I did not notice the character walking fast on Undertale compared to Deltarune. The Deltarune, when I pressed Shift, the guy walked fast, but Undertale it was not that fast. Maybe it's X for Undertale. I should try it. Yeah, so that's the whole such a good game, yeah. I'm really interested in how the story goes. UT. I don't know what that is. Ultimate Untitled what? Origin of replication. What does it does? Allows plastids to replicate independently. What is plastid? What is this? Masonic, can you explain what UT is? Or am I just stupid? Plasmid is a small circular double-stranded DNA molecule that exists separately from bacterial chromosome. Oh, Undertale. Okay, that's a good short form. Undertale. How long it took you guys to complete the game? I mean, on average, because one person told me that it took them 6 to 7 hours, and I think Google also said 6 to 7 hours. Found naturally in many bacteria like E. coli, replicates independently of the bacterial chromosome, carries extra genes, example antibiotic resistance, toxins, metabolic enzymes, not essential for basic survival, but useful under certain conditions. Never beat it, but I tried a long route. You still haven't beaten the game. Oh, I mean, is there a reason for not? And took me like 6 hours to get halfway of the long route. Okay, I don't remember. Okay, but I guess on average it's 6 to 7 hours. Let's see which route I take. I might go the long route. Plasmid is a small, not essential for basic survival, but useful under certain conditions. Plasmids are modified and used as cloning vectors to insert foreign DNA. Okay, I get the plasmid. So what does origin of replication do? Allows plasmid to replicate independently inside the bacterial cell. I got taken off of Game Pass. Okay, I get it. Well, I bought Undertale for €2. That should be equivalent of $2. May I ask what you mean by Deltarune's slow? How was Deltarune? I remember the Deltarune started with us being in the class, then going to get the chalk, and just and from there We met a Sans like guy, but it was not Sans. I forgot the name. And I don't know, maybe I was stupid because it took a long time for me to solve the puzzles. So maybe it's my issue that I felt the Deltarune a bit slow, but yeah, I did not feel a lot of story happening on the game. For Undertale, I met the woman, uh, I met the flower, the sunflower. That was good. This guy Sans. There's more characters in Undertale. At least I think Deltarune also gonna be having a lot of characters, but yeah. Selectable marker. What does it do? What it does? Allows bacteria to carry the plasmid to the selected. Usually provides antibiotic resistance. So, did you felt like Deltarune was slow, or did you feel like it was better? Which do you think it's better, Deltarune or Undertale? From what I heard, both are different games. That's what I heard. Like it's a different genre, or it's just different. Allows bacteria to carry plasmid. Allows bacteria to carry the plasmid to the selected. Could you explain more on that? Could you if proteins are they made let's go deeper and clear up how plasmids genes plasmid itself is a DNA, a circular molecule. It stores genes just like bacterial chromosome. These genes can code for proteins. Plasmid is not a protein, okay. uh, I personally think that's kinda unfair question, because since Deltarune hasn't released all the chapters yet. Okay, but so I'm guessing Undertale is just a single game. Deltarune has, I think, it has 4 chapters released, and the next one is coming up. Okay. Mm, do you think Deltarune is much longer than Undertale? I think it's a more hours. It takes more hours to complete Deltarune. The plasmid carries gene. Black codes for beta lactamase, a protein that breaks down ampicillin, plasmid DNA, mRNA, translation, transcription. I mean, you did not talk about selectable marker. What? Oh, for sure. Okay. So, which chapter are you on Deltarune right now? Did you get pretty far? Cause if I remember, the last thing I played on Deltarune was. Mm, I remember making a duck, a machine that could kill ourselves, and I made a duck. Yeah, the part where I felt a bit, I felt a bit slow was I had to fight these two people. Like, they were from our team itself, but they then broke and formed their own team or some shit. And I had to fight them, and they just won't lose. And if I lose, I thought it was a new story or new, they changed the story a bit. No, I just have to fight them again. I kept on losing. After two or three tries, I was like, am I supposed to fight them? Should I go for a new route? I don't know. I just felt a bit, I don't know what to do moment. Gene on plasmid that helps you to identify which bacteria actually took up the plasmid. What kind of gene usually codes for protein that gives antibiotic resistance? Selectable marker is a gene on plasmid. Examples of BLA gene, beta lactamase enzyme breaks down ampicillin. Is it made of mRNA? Yeah. Multiple cloning sites. It's a short sequence of DNA that contains many unique restriction enzyme sites. Does it code for anything? No. The MCS is non-coding DNA. It does not make proteins or mRNA is made from it. Why? It's where it cut the plastic open. Okay, it's inside the plastic. Okay. I completed all the chapters, but I'm planning to find secrets in chapter 3 and 4. Okay, you completed that part. Okay. So, do you know this part? I had to fight the purple kid and the skeleton person. Two of them I had to fight, and the skeleton person had a bike. The purple had an axe. I just keep on dying. Do you know how do you proceed this part? Do you have to just beat them? Because I felt like I cannot beat them. Okay, I get it. I get it. R. But no offense, but I feel like there's a skill issue on your part. Yes, you have to beat them. Okay, okay. I felt that. Okay, that makes sense. But I don't know. The Sans guy just don't lose health. That's what I remember. I'm curious where you are from. I'm from the United States of India. I'm from India. From specifically speaking, I'm from the southern part of India. Hell yeah. And right now I'm studying at a university in Österreich. That is Austria. It's a country near Germany. And if you know history, you know. Should I write it? I just read. At first, I just read them. So these are plasmids. Origin of replication is where the plasmid replicates itself independently. Uh, Selectable marker. It's where allows bacteria that carry the plasmid to be selected. Okay, multiple cloning sites. It's like an opening in the primer, plasmid. Sorry, plasmid and oh, cool. Yeah, it is. It's what it is. Well, I still don't get. I didn't understand selectable marker. What did I ask them? There's a gene on the plasmid that helps you identify which bacteria actually took up the plasmid. It usually codes for protein that gives antibiotic. What it? How does? How do we identify? I don't get it. I don't get it. If you feel like fighting isn't working, try acting. I did try, and they were not falling for it. The furthest I got with acting was I did make the purple one fell asleep for maybe 2 rounds, and that woman just woke up and start beating the shit out of me. How do we identify which bacteria actually got the plasmid? That's the heart of using selectable marker, and sometimes a reporter gene like LAC-Z. Let's break it down. Let's break it down. Insert your gene into plasmid. The plasmid contains origin of replication. That's where it copies itself. Selectable marker. I think that's the gene itself. So selectable marker is the gene that we are trying to replicate. Not quite. Common misunderstanding. Selectable marker helper gene tells you which bacteria. What do you mean, which bacteria? Gene of interest, the gene you're trying to replicate or study. Okay. Your plasmid might look like this: origin of replication, ampicillin, resistance gene, blah, selectable marker. Okay, selectable marker is like a tag or filter. So the genes you add onto the selectable marker repeats marker only starts. Dude, my spelling is fucked up. I don't know how to type at this point. Starts. It does not need your help or insert a gene to. Now, what happens when you insert your gene? You're not modifying the selectable marker. Instead, you insert your gene into a separate part of the plasmid, usually into the MCS region, the gene only expressed. It is placed under the control of promoter. The host cell has the right machinery. So to answer your questions, no, you don't add your gene into the selectable marker. You'd add your gene into the and think of plasmid like a house with security system. Selectable marker always working to keep unwanted bacteria out. A room where you put your custom furniture only shows up if you decorate it. Add promoter. Okay, so have you tried fighting the purple woman first and then fighting the sans guy? Okay, so you're telling me to kill, like defeat the purple woman, then go for the skeleton man. Okay, maybe. But the problem is the skeleton. I cannot skip the skeleton man. This guy rides his bike, and I cannot skip. I just cannot move my heart in a way that I don't take hits. I could. I need to practice. It's a skill issue. I get it. So to answer your question, no, you don't add your genes into a selectable marker. You add your genes into the MCS. Okay, so selectable markers are like tags, or set, or to be more precise, they are set of tags, set of nucleotides used to understand what genes lies. Selectable mark. Bro, bro, why you keep changing your thoughts? Mm-mm-mm. I got a message from a friend. So, selectable marker is a gene that produces a functional protein. So it's a gene with its own promoter. Okay, you need a promoter that produces a functional protein with a plasmid to survive. More specifically, type of molecule, a gene transcribed into mRNA, and it does not tell you what gene. It does not then what acts like a promoter for other genes, influence the inserted gene unless it's fused directly to it, which is not standard for a selectable marker. Analogy better one. Imagine you're entering a secure building. Selectable markers like the ID badge. Your inserted gene is what you're carrying in your backpack. Security, antibiotics, check if you have a badge. If yes, you're in the cell, survives. Oh shit, okay, I get it. So, selectable marker. Required for the plasmid to enter the nucleus of the bacteria. Excellent question, but here's the key. No. What? the Okay, so the prokaryotes don't have a nucleus, okay. So then what does the selectable marker do? Selectable marker is not involved in the entry, it is involved in after transformation. In screening or selection transformation, you introduce plasmids into the bacteria. Heat shock, electroporation. Some bacteria take up the plasmids, others don't. You grow all the bacteria on a medium containing an antibiotic. Only the bacteria that have the plasmid with the selectable marker gene will survive. The rest will die. Okay. So it's like a gene that gives permission for the resistance permission for the resistance protein to form, uh, which helps the bacteria to survive. Dude, my hand survive the anti antibiotic antibiotic what? Antibiotic situation. No. What's the word? Medium. I still didn't get it. Yes, selectable marker is a gene that produces a resistance protein which allows the bacteria to. So yeah, it's a real gene like any other. It has its own promoter, gets transcribed to mRNA, translated into a resistance gene. The protein actively destroys the antibiotic, so the bacterium lives. The bacteria without this gene die on the antibiotic plate. So what is the difference between the selectable marker and the gene that we are adding to the plasmid? I just don't get it. Since now I know you have gotten into that part of the delta room, what do you think of the whole lightener and darkener thing? Is it the part where I don't know, there's a tree and this thing circles and you have to keep following it until the it gets fully dark, and that's how the way is open? Is that what you're talking about? Because that was amazing. I was like, okay, but I don't know how you will figure it out. I did got some help from the stream. That's how I figured it out. Or else I'll just be there. So what exactly is the difference between the gene that is actually required to produce the protein and the selectable marker? The prophecy of Ralsai told. The prophecy Rasal. I don't know who that was on the stream. Was it Masonic 12 or I don't know. I don't remember who exactly was on that stream that day. Yeah, but it was from the stream that I could solve that puzzle. It's not functionally different in any structure from any other dreams. It just contains a promoter, a coding region, and it's transcribed to. But the difference lies in its purpose. Purpose helps to identify which bacteria has taken a plasmid. That so it's not okay. It's for us. It's not. It does not help the bacteria itself. It helps us to identify which bacteria. Ralsei is a character in the game. I don't think it was. Was it? Yeah, I think that the Ralsei is the one that's with us, right? The one with the magic and shit. I think that guy. But that guy did not tell me about. I should circle the cord. I mean, are you talking about this area in the game where there's uh there's this there's a black line that just keeps circling, and you have to keep following it so the whole area gets dark, and that's how you get to go to the next place. That's how you get to see a tree that is required for the next place to open. It's not functionally different in structure from any other genes. It still contains. So it is literally the gene that helps the bacteria to create the resistance. Codes enzyme beta lactase, which breaks down the antibiotic ampicillin, so bacteria with the plasmid, oh shit, survives. I'm talking about when you and the purple girl were talking to the hooded figure. Uh huh. Okay, I don't remember exactly this part. What exactly happens? I forgot. Does the screen just turns white and black? Is that what's happening? Cause I don't remember exactly anything special when the purple girl and the character met this Ralsei character. I think that's the prince, right? Ralsei is the prince or some shit. Okay, so I almost got it. Mm-hmm. So selectable marker is the gene on the plasmid that helps it to create antibiotic resistance to for its survival. Ampicillin resistance genes gene on a plasmid. What else? What else? Multiple a short DNA region where multiple restriction enzyme sites enables insertion of foreign DNA using enzymes. Located reporter gene helps detect the foreign DNA that has been successfully inserted. White colonies equals successful insertion. Blue colonies equals no insertion. Okay. Okay, this is how you. Okay. Interesting. Reporter gene. Nor I'll be right back in 10 seconds. 10, 9, 8, 7, 6, 5. You can feel it in the beat on a day like this is dream. It feel like summer. It feel like summer. Multiple cloning sites. What? Are these some restriction genes added to the added to what? The plasmid? Nor are you talking about the first time the main character and the purple girl talking to the Ralsai character? Is that what you're talking about? What is multiple cloning sites? No, it's not used to restrict genes. It's used to make it easier to insert genes using restriction enzymes. Chinese cradle points flat. Okay, so so it's like a set of codons. Yes, that's a great way to think of it. Let's redefine the sentence. Just likely to be 100%. You don't remember the prophecy? I do remember some of it. There is a king, and we have to talk to the king. The Ralsai is the son of this king. Is that how it is? That's what I remember. Is that how it is? I don't remember exactly. I'm sorry. My memory ain't the best. Multiple cloning sites is a short sequence of DNA, not exactly codons for translation, but rather specific nucleotides. I'm sorry, I meant nucleotides. That allows scientists to easily cut open. Yeah, choose an enzyme. Okay, so okay, I get the selective marker. I got the what was it? Multiple cloning sites. The reporter gene is just a gene that helps detect. You don't remember about the prophecy talking about a human, a monster, and a prince from the dark banishing the angels' heaven. Okay, now I remember some reading, going across something like this. Okay, a human, that's us. A monster, that's the purple, and prince, that's a raw. That's the one with the hood. Okay, banishing the angels' heaven. That's an interesting uh place to banish. It's a heaven. Now I think about it. First off, why are we studying in a place? Because when we started the game, we went to a school with monsters and humans. That's kind of sus now. Think when thinking about it. Where is it? Reporting engines. Uh, so if there is selectable marker, you don't actually need a reporter. Now I do remember this. The prophecy. Which sometimes scientists use different. Okay, so I got that one. What are the properties of reaction restriction enzyme? They cut open the multiple cloning sites. Oh, I could watch YouTube. Two minutes 30 seconds. That's it's not that big. Why is there ad? I'm pretty sure banishing the angels heaven is important since it's all about balancing light and dark. Okay. So when you got to the end of chapter 4, Did you get somewhere close to this angel's heaven? At least, did you see the angel? I mean,
Let's talk about. Hi guys, quick biochemistry basics here. Let's talk about restriction enzymes. Restriction enzymes are the enzymes that cleaves the DNA in small fragments. They're widely present in prokaryotes. They protect the DNA of prokaryotes against the invading bacteriophages.
bacteriophage.
Now you might wonder how come the restriction enzyme do not cleave the DNA of the host cell? The answer to this question lies in the fact that the host DNA is methylated at specific bases, whereas the DNA of bacteriophage is not methylated. And the restriction enzyme can fragment only non-methylated DNA. Hence, the host DNA remains protected. The restriction enzymes are classified into three types based on their recognition sequence and reaction requirements. Type 1 restriction enzymes cleaves far away about 1000 bases.
Nobody knows who's the angel to this day. Maybe we'll know in chapter 7. Okay, so did you had fun from chapter 1 to 4? Do you think it was
From the recognition sequence. They require ATP and SAM for their function. Type 2 restriction enzyme cleaves the palindromic sequence, which can be 4 to 8 base pairs in length. They require only magnesium ions for their function. Because of this reason, they are widely used in molecular biology.
let me just use my phone real quick.
Your palindromic sequence means the sequence which will read same when you read from both the direction. For example, type 2 restriction enzyme EcoR1 identifies palindromic sequence GAATTC. These
Oh,
enzymes often function as homodimers.
yeah, love the story. Okay.
Type 3 restriction enzymes. These enzymes recognize two separate non-palindromic sequences which are inversely oriented. The type 3 restriction enzyme cleave 20 to 30 base pairs after the recognition site.
Why ads? I still didn't get it fully.
In this video, I will be discussing
Love
restriction
this story.
endonucleases. Restriction endonucleases, also called restriction enzymes, are enzymes that cleave DNA at
So,
specific
Noor,
sequences.
have you played any other
These
games?
enzymes
Oh, okay.
are
I
particularly
think you're
valuable in a lab setting.
right now you're playing Deltarune, right? You wanna complete all the achievements.
In this video, I will be discussing restriction endonucleases. Restriction endonucleases, also called restriction enzymes, are enzymes that cleave DNA at specific sequences. These enzymes are particularly valuable in a lab setting because they allow scientists to cut, isolate, and manipulate DNA segments of interest. Restriction enzymes are naturally found in bacterial cells. These cells use their restriction enzymes to cut apart DNA that has been injected into them by viruses. Cutting up the viral DNA prevents the virus from successfully reproducing within the bacterial cell. The key is for the restriction enzymes in the bacterial cell to recognize sites that are only found in viral DNA and not found in the bacterial DNA. The bacteria wouldn't want its own DNA to be cleaved by restriction enzymes. So as I said,
didn't get it. What?
Bacterial cells. Restriction enzymes are naturally found in bacterial cells.
Okay.
These cells use their restriction enzymes to cut apart DNA that has been injected into them by viruses. Cutting up the viral DNA prevents the virus from successfully reproducing within the bacterial cell. The key is for the restriction enzymes in the bacterial cell to recognize sites that are only found in viral DNA and not found in the bacterial DNA. The bacteria wouldn't want its own DNA to be cleaved by restriction enzymes. So as I've said, the restriction enzymes can only recognize and cleave DNA at specific sites. This site is called a restriction site, which is often 4 to 12 bases in length.
I've been also playing some Baldi's Basics. I heard of the game, Baldi's Basics. I'll look it up right after this video.
Enzymes can only recognize and cleave DNA at specific sites. This site is called a restriction site, which is often 4 to 12 bases in length. For example, the restriction enzyme EcoR1 recognizes and cuts DNA at this site. It will scan the DNA, and when it finds the site GAATTC, it will cleave it like this. You'll notice that this cut does not go straight through the two strands. Instead, it creates what is called sticky ends, where these 4 bases are left without their complementary partner. Having sticky ends makes it easy to attach other DNA molecules that have been cut by the same restriction enzyme together. So we can see here that this molecule has 2 restriction sites. It will get cleaved in these sites by the restriction enzyme, and now we have 3 separate molecules of DNA. This second molecule of DNA has had a substitution mutation in the second restriction site. This means that the restriction enzyme would not cleave this molecule here. Instead, it would only cleave here, creating 2 separate fragments of DNA. So in a lab setting, what do you do after you've cleaved a DNA molecule? You can separate these DNA molecules using gel electrophoresis, and then after separating them, isolate a piece of interest. You can take this DNA piece of interest that you've isolated and insert it into a plasmid that's been cut by the same restriction enzyme, and then you can insert this plasmid into a cell to transform bacterial cells. All in all, some pretty cool things as a result of a restriction enzyme cleaving DNA in specific locations. If you're interested in learning more about what happens in a lab after you use restriction enzymes, check out my video on gel electrophoresis or my video on transformation.
I do remember these stuff learning in school. I've been playing some Baldi's Basic. Have you ever heard of it? I think I think weird would be the best way to describe it. Oh, now you got my attention. Baldi's basics. Oh no, Baldi. There's one. Badlies. Okay, bad. No, Baldi's. Okay. Yeah, I seen this game before, and yeah, it's a weird game. I don't know why, but it just feels so weird. Oh, it's used for learning. Okay. No Gemini. Really? So, is it an educational game? Okay. And oh, by the way, every character in Deltarune has a secret boss, at least as of now. Every chapter, okay. So there's one secret boss per chapter. That's good. So four secret bosses. Did you clear all the four, or are you on your way to pursue them? I pressed enter. Why? Basic math problems, school rules and discipline, escape survival, the lore, fan theories. Okay. I don't know, looking into an AI for a game advice, but I haven't completed chapter 3 and 4 yet. Okay. So you did do chapter 1 and 2. That's good. Now, what are the properties of restriction? Specific recognition sequence. Most recognized short palindromic sequence. E. coli, echo R1 cuts GAATTC. Some recognized degenerate or non palindromic sequence. Types of restriction enzymes cut DNA from far from recognition site. Requires ATP type 2. Most commonly used cut within near recognition sites do not need ATP. Oh, that's interesting. Type 3 cut 25 base pairs away from recognition sites requires ADP. Cut methylated or modified DNA. Cleavage patterns. Blunt ends cut strands at the same position. Sticky ends create overhangs. So some enzymes are blocked by DNA methylation. Bacteria protect their own DNA methylation. Optimal reaction condition. Some enzymes are blocked by DNA methylation. Bacteria protects their own DNA through methylation. Optimal, what are the properties? Optimal reaction conditions: pH, salt concentration, temperature, star activity. Also, yeah, I also play a game called Lice of P every day, 15 minutes. That would be the other game that I play other than Undertale or Deltarune. What? Triple point of water. I got 31 subscribers. Yeah, yesterday was 29 and today it passed 30. Let's go. Let's fucking go. That's amazing. Just imagine 30 people just went in like, yeah, I support this guy. I'm happy. I'm happy to be here, bro. I'm happy to be here. Or sister, bro, or sister, doesn't matter. I'm happy to be here. Most recognition sequence are 6 base pair or 4 base pairs long. The cut can open either central, creating gene regulation in lack operon of E. coli. Let's see what we got.
If you're a first or second year med student taking anatomy and you're feeling a little bit overwhelmed because of how much information there is to
Nope.
learn,
Hey everyone, quick biochemistry basics here. So let's talk about the lacoperon. The lacoperon is the best studied model for
Lacoperon.
understanding the regulation of gene expression. The genomic DNA of bacteria has many genes. Genes, by the process of transcription and translation, forms a protein. Both transcription and translation are highly energy intensive, hence only those genes that are required for homeostasis are continuously expressed. This type of expression is known as constitutive expression. Most of the genes are controlled by the extracellular signals, which are usually molecules in the growth medium. The signals that positively regulate the gene expression are called activators, while the signals that negatively regulate gene expression are called repressors. Both activator and repressor are the DNA binding proteins that regulate the gene expression. The DNA sequence at which the repressor binds is called an operator, while the DNA sequence at which the activator binds is called an activator binding site. Now let's talk about the lacoperon. An operon is a functional unit of DNA containing cluster of genes under a single promoter. The lacoperon has three genes, LAC-Z, LAC-Y, and LAC-A. The LAC-Z gene encodes beta-galactosidase enzyme. This breaks lactose into glucose and galactose. The LAC-Y gene encodes lactose permease enzyme. This enzyme transports lactose inside the cell. The LAC-A gene encodes thiogalactoside transacetylase enzyme. and it helps in the removal of toxic thiogalactosides, which are also transported by the LAC-Y. The LAC-I encodes a repressor that binds the operator and represses the lac-operon. Let's see how the lac-operon functions. In absence of lactose, the operator is occupied by lac repressor. This ultimately shuts down the lac-operon genes. Basal level transcription. Now, sometimes, even in absence of lactose, the repressor may fall off from the operator, allowing the transcription of lac genes.
Well,
This occurs
that's
because the binding
pointless.
of repressor with the operator is not permanent. Because of basal level transcription, there is always some amount of lactose permease, beta galactosidase, and thiogalactoside transacetylase enzyme present in the cell. When lactose is present, lactose enters the cell with the help of lactose permease. Remember, lactose permease is present because of the basal level transcription of the lac genes. The lactose that enters the cell binds with the repressor and releases the repressor from the operator. As the operator is free, the RNA polymerase can now bind the promoter and initiate the transcription of lac genes. Now, as lac genes are continuously transcribed, more amount of lactose permease, beta galactosidase, and thiogalactoside transacetylase enzymes are formed. The lactose permease further allows more amount of lactose to enter the cell. The lactose that enters the cell is hydrolyzed to glucose and galactose by beta galactosidase enzyme. Glucose and galactose are used as an energy source. The role of CAP protein in lac-operon. CAP stands for catabolite activator protein. CAP binds CAMP, hence it is also known as CAMP receptor protein. When the glucose levels in the cell decreases, the level of cyclic AMP in the cell increases. The CAMP binds CAP protein and activates it. The CAMP CAP complex then binds to the CAP site and interacts with the C-terminal domain of the alpha subunit of RNA polymerase enzyme. This increases the rate of transcription of lac genes.
That's it.
Willst du wissen, wie YouTube...
Hmm. The gene regulation of lac-operon was first genetic regulatory mechanism to be understood clearly, so it has become a foremost example of prokaryotic gene regulation. This lactose metabolism system was used by Francois Jacob und Jacques Monod, interesting names, to determine how a biological cell which enzyme to synthesize their work on lac-operon won them the new Nobel Prize in physiology. So I kind of get the lac-operon. Describe blue-white screening in molecular cloning. So I believe that's the sep. I forgot the name. I bet, or I just think that's this guy. Where is it? That's the reporter gene. So Noor, are you still up here? I'm sorry, man. I was just looking at different topics. Yeah, and the exam is tomorrow. I don't think I'll pass the exam tomorrow because I still need to learn more. But hey, the more you know, the better you get.
Hello and welcome to today's episode about the blue-white screen. We will explain what it is and how it is performed. Let's start off with some basic things like transformation. What is transformation? DNA transformation is the process during which a plasmid or vector carrying a specific gene of interest is transformed into another cell, often bacteria. We can see here we have a plasmid, which is a circular bit of DNA, and
The
in
fuck?
orange we can see the gene of interest inserted in the plasmid. The most common used bacteria for this process is E. coli. What happens if we transform a plasmid into bacteria like E. coli? We can insert this plasmid via heat shock, for example, and then the gene of interest is
Oh.
transcribed and translated into proteins. This was just a little short explanation of transformation, and we need this to explain the blue-white screen. The blue-white screen is a screening technique which is one of the most common used techniques for detecting recombinant bacteria. It is used to check whether the transformation process was successful or not. We are going to start off with a normal bacterium. So here we see the genomic DNA on the right and the plasmid DNA, which was inserted via heat shock, for example, on the left. And for the blue-white screen, we used the fact that one of the proteins or enzymes is built up
I'm gonna
from two
try to beat
different
the chapter
types of
three's boss.
subproteins.
Bye. You can do it, man. Peace out. I really appreciate Nor for checking out the stream. Good luck on the boss, man. See ya. I really appreciate you for checking the stream.
One is usually transcribed and translated from the genomic DNA of the E. coli, and the other one, the alpha unit, is only transcribed from the plasmid. And only together they form the active enzyme called beta-galactosidase. Here on the right we can see a prepared plate. This plate contains X-gal. And on X-gal plates we can see blue dots, and these represent colonies of bacteria which have this enzyme called beta-galactosidase. If there is no functional beta-galactosidase, we see only white colonies on the X-gal medium. This is just the background information to understand the upcoming design of the plasmid. We have a plasmid here, and in orange you can see the ampicillin resistance. We usually work with the ampicillin resistance because ampicillin is an antibiotic, and with the gene that allows the bacteria to live on ampicillin medium, we can know, okay, these bacteria, they contain the plasmid. And this is the first thing we should know. The yellow sequence on the plasmid represents the LAC-Z gene. And this LAC-Z gene will produce functional beta-galactosidase later. These are the two main components of our plasmid. But now we have to make sure that we also detect the restriction site. This plasmid is designed so that we have a restriction site at the end, but in the sequence of the LAC-Z gene. In biotechnology, we can use restriction enzymes to cut specifically at the restriction sites. Here you can see during the cut, a part of the LAC-Z gene will go missing. The DNA insert is designed so that it can easily fit into the restricted site. Once the DNA insert is inside the plasmid, we call it recombinant DNA. And as we said earlier, now the sequence of the LAC-Z gene is now not completely present. This was due to a part that went missing during the restriction. So we conclude, we have our plasmid now, this carries a ampicillin resistance, and we have no LAC-Z gene anymore, and we have the inserted DNA, and we can call this plasmid recombinant DNA. We remember from the introduction that if we place bacteria that have a functional LAC-Z gene, a functional beta-galactosidase, on X-gal plates, we should expect blue dots. But since the Laxet gene is missing here, what we will see is white colonies. In the laboratory, in real life, a screen would rather look like this. We have a plate, this contains egg gall and ampicillin, making sure that only bacteria with a plasmid will survive. And then we will find blue dots and we will find white dots. And these dots represent the colonies of bacteria. A white colony means we have successfully made recombinant DNA. A blue colony, on the other hand, means there is no recombinant DNA. And now we can selectively use the white colonies and place them on a different plate and work with them, since we know our DNA insert should be contained on the plasmid. Thank you very much for watching. I hope this little introduction to the blue-white screen was helpful for you. If it was,
Okay, so
please
it's
give this
just to
video a like,
understand. Yeah, it's
and I
similar
hope
to
we will
the.
see each other next time.
It's similar to
They don't
the
believe how toxic this is. If you're still using a plastic or
reporter
wooden cutting
gene.
board,
There are some pieces of technology you really develop an appreciation for. Maybe it's your phone, your laptop, your drawing tablet, but for me, it is the copy machine. Oh, the copy machine. I know it's not possible, but I could promise you that whenever the copy machine was aware of my presence, that's when it decided to malfunction. Especially if it knew I was short on time. And of course, when it jams, the fancy copy machines will even tell you what is wrong and how to fix it. But my experience with trying to follow the instructions of fixing the copy machine makes it worse. And then by that point, there's a line, and you feel your heart pounding, and you're embarrassed because now you've jammed it for everyone. Yeah, so while I've developed an appreciation and respect for the copy machine, I've definitely tried to limit my use of it over the years. Saves paper anyway. You may be wondering, why is she spending so much time on this? Well, I want to call your attention to a technology, a biotechnology, that works almost like an amazing and fancy copy machine. Except not for something on paper. No, it's for DNA. This biotechnology is PCR, polymerase chain reaction, or PCR for short. It provides a way to make more copies of a portion of DNA. Lots and lots and lots of DNA copies, and this technology does not need to happen in a cell either. In fact, it can happen in a test tube. And so you may wonder, number one, how does PCR work? And number two, why? Why make more copies of some specific portion of DNA? So let's briefly answer those two questions. To answer the first question, the how does this work question, let's talk about what we need first before we get to the steps of how it works. We need the DNA portion that we want to make copies of. We need some kind of buffer to put it in, and then we need things that would be necessary to make more copies of the DNA. So think about what that might mean from our DNA replication video. We'll need primers. Recall that primers help DNA polymerase, a building enzyme, know where to go to start its building. We need DNA polymerase, the building enzyme. Fun fact, the DNA polymerase used is often a heat-resistant type of DNA polymerase, as PCR uses heat. Typically, the polymerase chosen for the job is Taq polymerase, a type of heat-resistant DNA polymerase. Taq polymerase is originally from a type of bacteria that can handle and live in really high temperatures in nature, in hot springs. Pretty cool. We also need DNA nucleotides for the DNA polymerase to build with. So we can look at the PCR sequence in three major steps. We'll illustrate with one double-stranded DNA molecule here, and let's assume this is
Well, people, I have to head to work in 1 hour, and I have to reach the bouldering place at 2. So it takes about 30 minutes for me to get from here to there. So I should leave at 1:30, and I need to eat something before I head out for work and bouldering. So I guess that's it for today. And I am not being serious. We still have tomorrow. We do this, bro. We do this. Well, peace out, people. I today I had a lot of people come to my stream. I really appreciate it, people.